Concept 16.1, DNA is genetic material. In this concept, we will be discussing DNA versus protein as the genetic material of cells and the structure of DNA, including base pairing. The first part, DNA versus protein, covers a lot of history, but it shows how the process of science works to show how the natural world operates. It may seem that DNA is obviously the molecule that carries the genetic information of the cell, but this is hindsight. When we were figuring out which macromolecule carried the genetic information, protein was the front runner. It can fold into shapes that can do anything in the cell, and there's more protein in a cell than any other molecule. Carbohydrates and lipids, although found in high quantities in the cell, are limited in their shapes and were not considered as the carrier of genetic information. Nucleic acid is found in minuscule amounts in the cell. Because of ATP, we knew that nucleic acid carried energy around, but how could it carry genetic information? A group of scientists led by T.H. Morgan showed that genes exist as parts of chromosomes, and chromosomes are composed of DNA and protein. So which was it, DNA or protein? Two groups helped cement DNA as the genetic material of the cell: Frederick Griffith and Hershey and Chase. Frederick was working on a vaccine for pneumonia caused by Streptococcus pneumoniae. There are two main strains of S. pneumoniae. One is non-virulent, or it doesn't cause disease and it doesn't have a capsule or sugar coating that prevents the immune system from swallowing it down. The other strain does have a capsule and it does cause disease. If he injected the cells with the capsule or S strain, the mouse would die. Not a good candidate for a vaccine. Mice injected with the capsuleless R strain didn't get the disease. They were just fine. They didn't get pneumonia, but they also didn't get very good immunity against the S strain. He tried injecting mice with heat-killed S cells. That worked pretty good, but he also tried mixing the live R strain with the heat-killed S strain. These mice died. When he tested the blood of the dead mice, he found live S strain cells. Where did they come from? Were they zombie cells? Undead cells rising from the grave to plague mice kind? Well, no. Sounds like a good plotline, though. He determined that the live cells must have picked up material that coded for the capsule from the dead, lysed S cells. But what was that material? DNA or protein? Another group led by Avery indicated that it had to be DNA rather than RNA or protein. A later experiment by Hershey and Chase confirmed this. But first, a bit about viruses of bacteria, or bacteriophages. Phages, as they are known, have just two macromolecules, DNA and protein. They infect bacteria, taking over the bacterial cell machinery to make more phages. Part of the phage remains outside of the cell. This part looks kind of like a lunar landing module. Something has to go into the cell to take it over. So Hershey and Chase took two groups of phages and radio-labeled one group's protein and the other group's DNA. After allowing the phages to infect the bacteria, they separated the phages on the outside from the infected bacteria. They only found radio-labeled protein on the outside of the infected cell. They only found radio-labeled DNA on the inside of the cell. This confirmed Avery's results. Now that we knew that DNA was the genetic material, we needed to figure out how it passed on information. The first step to do that was to figure out the structure of DNA. Remember, there isn't a lot of DNA in a cell to work with, so that was kind of a big deal. There were two groups working on the structure of DNA during the early 1950s, but we will focus on two: Watson and Crick, and Franklin and Wilkins in London. The textbook has a more detailed summary of how Watson and Crick eventually came up with the structure of DNA, and makes it sound like the two labs were working together amicably. The reality shows that scientists are human too. Franklin and Wilkins were not getting along. Both had understood that they were to be in charge of the lab. Rosalind Franklin, rather than continue to argue with Maurice Wilkins, got to work doing what she'd been hired to do, which was to perform X-ray diffractions on crystals of DNA. This was her specialty. From this, she was able to determine that DNA had two strands that twisted around each other in a helix, and that the sugars and phosphates were on the outside, and the two strands were equal distance apart. Meanwhile, Wilkins became buddies with Watson and Crick, and showed them some of Franklin's unpublished work, including this X-ray diffraction photograph. They came to much the same conclusion that Franklin did. Rather than doing X-ray diffraction, they were building models to visualize what structures would work. They combined what they had learned from the X-ray diffraction photograph and some early work by a researcher named Chargraf. Chargraf had discovered that DNA has the same ratios of A and T versus G and C. A and G both have 2 rings in their bases. T and C only have 1 ring in their bases, making them smaller than A and G. If A always hydrogen bonds with T, like in this figure, they are the same distance apart as G and C when they hydrogen bond. That explains Chargraf's rules. Putting the bases on the inside also explains what they had seen in the X-ray diffraction pattern. Eventually, Watson, Crick, and Wilkins shared the Nobel Prize for the discovery of the structure of DNA. Unfortunately, Franklin had died of cancer by that point, and Nobel Prizes aren't awarded posthumously. Let's review base pairing in DNA. Adenine, or A, always hydrogen bonds or base pairs with thymine, or T. Guanine, or G, always base pairs with cytosine, or C. As you can see, A and T form 2 hydrogen bonds. G and C form 3 hydrogen bonds. If G tried to bond with either A or T, it would be awkward, since you can't form 2.5 hydrogen bonds. If G tried to bond with A, the strands would be further apart than when it bonds with C, making a bump in the line of the sugar phosphate backbone. Keep that in mind when we discuss proofreading during DNA replication. Well, that's it for this concept. Make sure you bring any questions you may have to lecture.